Welcome to the deep dive. Today we're unlocking the secrets of a system that is quite literally orchestrating your entire existence right now, often without you even realizing it. So to kick us off, let's talk about this case. A 56-year-old woman, her world had just shrunk dramatically. Her main complaint, loss of vision. But here's the thing, her eyes themselves, perfectly healthy. The actual problem was her eyelids. They were closing involuntarily. With such force and frequency that she couldn't really see her surroundings for more than these brief, fleeting moments. It's a condition called blepharospasm, and it was completely debilitating for her. Think about it. She'd lost her driver's license, was falling at home. It was a really life-altering challenge. Now, here's where it gets, well, truly remarkable. The treatment involved several tiny injections of a really potent toxin botulinum toxin type A, just into a small muscle around each eyelid. And within only 2 days, she reported this dramatic improvement in her vision. Just think about that for a second. Using a toxin as a kind of precision medicine to restore sight. It seems incredibly counterintuitive, doesn't it?
It absolutely does. And this captivating case, it isn't just a story about one specific treatment, for involuntary muscle spasms. It's actually the perfect entry point for understanding one of the real unsung heroes of our physiology, the autonomic nervous system. The ANS.
Okay, yeah, let's unpack this then. Today, we're going to deep dive into the ANS, this incredibly complex, largely unconscious control system. It orchestrates, well, basically everything, from your heart rate and digestion to even subtle immune responses. Our goal here is to give you, our listener, a truly elite professional understanding of this vital system. We want you to master its intricate anatomy, its sophisticated chemical language, and precisely how powerful drugs can influence it.
Exactly. And as our foundational map for this whole exploration, we're drawing from some pretty comprehensive source material, specifically key excerpts from KATZUNG's Basic and Clinical Pharmacology 16th ed. So this ensures we're really delving into the absolute depth and accuracy of the information needed for that kind of mastery they're talking about.
Great. So let's start by looking at the grand blueprint, the entire nervous system. At the broadest level, it's kind of elegantly divided into two main parts, right? First, the central nervous system, the CNS. That's your brain and spinal cord, the body's central command center. And then basically everything else, all the neuronal tissues outside the CNS, that falls under the peripheral nervous system, or PNS.
Precisely. And it's within that peripheral system, the PNS, that we find this crucial functional division. You've got the somatic nervous system, or SNS, and the autonomic nervous system, the ANS. Now, the SNS is what you consciously control. Think about reaching for your cup, walking, even just maintaining your posture right now. Its efferent or motor portion is all about those, voluntary, intentional actions. The ANS, on the other hand, that's the autonomous one. It operates largely independent of your direct conscious control, working tirelessly behind the scenes. Its main role is to control and integrate all those vital, visceral functions. We're talking about essential life-sustaining processes. Things like your cardiac output, how blood flow is precisely distributed throughout your body, the entire digestive process. It's truly the hidden maestro of your inner world.
Okay, here's where it gets really interesting and maybe a bit surprising for some listeners. While the ANS is mostly known for controlling these automatic bodily functions, evidence is really accumulating that it has some much broader, more surprising influences. For instance, the vagus nerve, a major highway of the ANS, right? It isn't just controlling your heart and gut. It can influence your immune function, even modulate CNS functions like seizure discharge. And remarkably, there's even evidence now indicating that autonomic nerves can influence cancer development and progression. It's a system that really touches nearly every corner of your biology.
And it's definitely not just a one-way street of command signals going out. Both the autonomic and the somatic systems relied heavily on crucial afferent or sensory inputs coming in. These inputs provide vital information about both our internal state, what's going on inside, and the external environment. This incoming information then constantly modifies the motor output through various reflex arcs, allowing your body to adapt and respond, seamlessly.
It makes sense. And when you think about the body's major regulatory systems, the nervous system shares some fascinating common properties with the endocrine system, the body's hormonal network. Both involve this high-level integration in the brain, both influence distant regions of the body, and both make extensive use of negative feedback loops.
Exactly. It's like they're both working constantly to maintain that perfect internal balance, that homeostasis. And beyond just electrical signaling, both systems also heavily rely on chemical transmission. In the nervous system, this happens when tiny amounts of these special chemical messengers, we call them transmitter substances, are released. They come from the ends of nerve terminals into this microscopic gap called the synaptic cleft. These transmitters then just diffuse across that gap and either activate or inhibit the next cell in line. by binding to specialized receptor molecules on that cell. It's like a beautifully precise chemical handshake, allowing for really rapid and targeted communication. And interestingly, in a few rare instances, we even see what's called retrograde transmission. That's where a signal actually goes from the receiving cell back to the sending neuron, subtly modifying its future activity. So it can be a two-way street.
Fascinating. So what does all this mean for us, especially thinking about pharmacology? It means that by either mimicking or blocking these specific chemical transmitters, we can selectively modify a huge array of autonomic functions. This is exactly why autonomic drugs are so incredibly useful in treating clinical conditions. But it also explains why many drugs used for other things, like allergies or mental illness, can have those often unwanted side effects on our autonomic functions. These powerful chemical messengers act on a whole variety of effector tissues: cardiac muscle, smooth muscle, even the vascular endothelium, exocrine glands, and, interestingly, other presynaptic nerve terminals too. It creates this complex web of influence. Okay, so now let's take a closer look at the actual anatomic divisions within the ANS itself. We generally talk about two main ones: the sympathetic, often called the thoracolumbar division, and the parasympathetic, which has traditionally been referred to as the craniosacral division, though we'll have to update that second one a bit. Right, let's trace the paths of these nerve fibers, specifically the preganglionic efferent fibers. These are the ones starting in the CNS and going out to those intermediary relay stations, the ganglia. In the sympathetic division, these fibers exit the CNS through the thoracic and lumbar spinal nerves, and as we'll get into, maybe even through newly recognized sacral spinal nerves too. They're typically quite short, these preganglionic fibers. They terminate in what are called the paravertebral chains, ganglia running alongside the spinal column, or sometimes in prevertebral ganglia, which lie out further on the front surface of the aorta. From these ganglia, longer postganglionic sympathetic fibers then travel out to their target tissues. Now, for the parasympathetic division, the preganglionic fibers originate way up in the brainstem, exiting through specific cranial nerves, numbers 3, 7, 9, and X, and also from the sacral region of the spinal cord. These fibers are generally quite long, unlike the sympathetic ones. They terminate either in distinct peripheral ganglia, like the ciliary or submandibular ganglia, or, very commonly, they extend much further out. Sometimes they even diffuse directly within the walls of the organs they innervate, with their ganglia located extremely close to the target cells.
Okay, this brings us to a really crucial, almost paradigm-shifting insight. It challenges some long-held beliefs, and it's definitely a deep dive highlight for us today. The sympathetic sacral outflow reclassification. For a very, very long time, it was just accepted wisdom that the cholinergic nerves innervating pelvic organs, think rectum, bladder, reproductive organs, were part of the parasympathetic system, just like the cranial nerve cholinergic system. However, recent, really compelling evidence now strongly suggests that these sacral cholinergic fibers are, in fact, sympathetic. They seem to have originated from embryonic sympathetic precursor cells.
Wow, okay. That's a big shift. What's the evidence for that?
Well, the lines of evidence are quite compelling, actually. First, if you look at the genetic blueprints, the gene expression of these neurons, cranial parasympathetic neurons express specific developmental genes and transcription factors, things like FOX2B, TBX20, TBX2, TBX3. But interestingly, thoracic sympathetic neurons and these sacral preganglionic neurons do not express those specific genes. Instead, the sacral neurons uniquely express something called FOXP1, which the cranial ones don't. So their genetic programs are different from the start.
Different wiring instructions, basically.
Yeah, exactly. Second, look at where they exit the CNS. Cranial parasympathetic fibers exit via dorsolateral points in the brainstem, but sympathetic and these sacral preganglionic nerves exit the spinal cord via the ventral root exits, a totally different pathway out. And third, even during early development, their patterns of expressing specific transporters and enzymes are distinct. It all points away from them being truly parasympathetic. These observations together build a really powerful case for changing that traditional craniosacral label for the parasympathetic system. It's probably more accurate, anatomically precise, to call it the cranial autonomic nervous system now. This is a pretty big deal in our fundamental understanding of how the body's wiring actually works.
That is a monumental reclassification, and it really drives home the point that Sympathetic and parasympathetic are, at their core, anatomic labels, right? They aren't defined by the type of chemical transmitter released or whether the effect is excitation or inhibition. It's all about where the nerves originate and where they go.
Absolutely. And we must always remember the crucial role of those afferent fibers running back from the periphery, back to integrating centers like the enteric plexuses, the autonomic ganglia, and the CNS itself. These sensory inputs provide that constant real-time information leading to complex reflex motor activity. And what's also fascinating is that we're increasingly finding some of these sensory fibers also have peripheral motor functions themselves. It's a true feedback loop.
Okay, moving deeper into the periphery, let's talk about the enteric nervous system, the ENS. This is often called the third division of the ANS, and it's just massive. We're talking over 150 million neurons.
Yeah.
That's a lot of specialized brain power just for your gut.
It really is incredible. The ENS is this elaborate neural network embedded right within the walls of the entire gastrointestinal tract, basically from your esophagus all the way down to the distal colon. It's deeply involved in both the motor activity, the contractions, and the secretory activity of the gut. It plays a particularly significant, often independent role in things like colon motility. It has two main layers, or plexuses. The myenteric plexus, also known as Auerbach's plexus, which sits between the muscle layers, and the submucous plexus, or Meissner's plexus, found closer to the inner lining. These intricate networks receive input from both parasympathetic preganglionic fibers and postganglionic sympathetic axons. But crucially, they also get direct sensory input from within the gut wall itself. Fibers from these plexuses then branch out in all directions to control the gut's smooth muscle contractions and its secretory cells.
And the sensory fibers within the ENS itself, they're pretty remarkable too, aren't they? They're constantly transmitting chemical information, mechanical information like stretch from the mucosa and stretch receptors back to motor neurons within the plexuses and also out to postganglionic neurons in the sympathetic ganglia. For instance, these tiny endocrine epithelial cells in your gut lining can actually synapse with vagal and other sensory fibers. They use chemical signals like glutamate for really rapid transmission of information about, say, the chemicals in your gut contents back to the CNS. And then they might use peptides like cholecystokinin for slower, longer-lasting kind of modulatory effects.
Right. And it's important to clarify that while the ANS provides crucial input to the ENS, this input is primarily modulatory. It influences it, but doesn't necessarily run the whole show. Even if you completely cut off ANS input to the gut, perhaps through injury or certain conditions, core GI activity isn't abolished. It keeps going. In fact, sometimes selective denervation can even enhance motor activity, which really showcases its remarkable ability to function as a semi-autonomous system, like a little brain in the gut.
That's what's truly fascinating here and really essential for that elite professional understanding we're aiming for. How the ENS functions semi-autonomously. It integrates information locally, it modulates GI activity using that ANS input, it sends vital sensory information back up to the CNS autonomic centers, and crucially, it ensures synchronized impulses. This synchronization is what makes sure, for example, that gut contents are propelled forward efficiently, not backward, and that sphincters relax precisely when the gut wall contracts. It allows for incredibly sophisticated independent control right there in the gut wall.
Okay, so we've talked structure divisions. Now let's turn our attention to the actual chemical language these nerves use. How do they communicate at the cellular level? When we look at tight classic synapses, like the neuromuscular junction where a nerve meets a muscle, or most connections between neurons, the nerve terminal ends extremely close to the cell it's talking to. This means the diffusion path for the chemical messenger, the transmitter, is very short, and that leads to rapid, highly localized effects. Very precise.
Right, a direct hit. But that's different from many autonomic junctions, isn't it? Where the nerve talks to smooth muscle or cardiac muscle or glands. There, the transmitter is often released not from a single tight spot, but from this chain of varicosities, like beads on a string along the nerve fiber. And the gap, the junctional cleft, is often wider too.
Exactly. That setup leads to a slightly slower onset of action, and it can activate or inhibit a broader range of effector cells at the same time. It allows for a more diffuse yet still targeted influence across the tissue. Now, traditionally, we classify autonomic nerves based on the main chemical messenger, the primary transmitted molecule they release. So we have cholinergic nerves, which release acetylcholine or ACE, and we have noradrenergic nerves, which release norepinephrine or NE. NE is also sometimes called noradrenaline, so these are also called adrenergic nerve.
Okay, cholinergic and noradrenergic NE, where do we find each of them?
Well, cholinergic fibers, the ones releasing ASHAC, are incredibly widespread. They include all preganglionic efferent autonomic fibers. That means pretty much every nerve fiber leaving the CNS to go to a ganglion uses ASHEC. They also include the somatic motor fibers that go directly to your skeletal muscles, making them responsible for voluntary movement. Plus, most parasympathetic postganglionic fibers are cholinergic. And interestingly, some sympathetic postganglionic fibers are too, like the ones that innervate your sweat glands. It's also worth noting some parasympathetic postganglionic neurons aren't just using ASHEC. They might also use other messengers like nitric oxide or various peptides, either as the primary transmitter or as car transmitters, adding another layer of complexity.
Okay, so ASHEC is really common. What about NE, the neurotransmitter fibers?
Noradrenergic fibers, primarily releasing NE, make up the majority of the postganglionic sympathetic fibers. While dopamine is a crucial transmitter in the CNS and might be released by a few peripheral sympathetic fibers, the main player out there is NE. And then you have the adrenal medulla, which is embryologically like a modified sympathetic ganglion. It gets a direct sympathetic preganglionic input, using AMTAU, remember, and releases a powerful mix of epinephrine, adrenaline, and noradrenaline directly into your bloodstream. This acts like a body-wide hormonal surge, part of that fight-or-flight response.
Right, this detailed understanding of the chemical communication, it brings us straight to a really profound insight for pharmacology, doesn't it? When we think about how drugs work, there seem to be 5 key features of neurotransmitter function that act as the precise targets. You've got the synthesis of the transmitter, its storage inside the nerve, its release into the synapse, the termination of its action once its job is done, and finally, its receptor effects on the receiving cell. Understanding these 5 steps seems absolutely fundamental to mastering how we can intervene and modify autonomic function. So maybe let's take acetylcholine, that key player from our opening story, and really unpack how it goes through each of those steps.
Absolutely. Let's delve into the intricate choreography of cholinergic transmission, hitting those 5 features. First, synthesis. Acetylcholine is built right there in the nerve cell's cytoplasm from 2 basic ingredients, acetyl-CoA and choline. The enzyme doing the building is called choline acetyltransferase, or CHAT. Now, crucially, that choline ingredient has to be brought into the neuron from the outside fluid. This happens via a specific sodium-dependent membrane transporter called CHT. And interestingly, this transporter can be blocked by certain research drugs called hemicolinium. So blocking uptake blocks synthesis. That's step 1.
Got it. So it's made, then what, storage?
Exactly. Storage. Once synthesized, ACH isn't just floating around loose, it's carefully transported from the cytoplasm into these tiny protective delivery bubbles called vesicles. This transport is handled by another specific protein, a vesicle-associated transporter, or VAT. It's powered by proton movement and can be blocked by a research drug called Vesamicol. AZ gets packaged in these discrete packets, or quanta, each vesicle holding anywhere from, say, 1,000 to 50,000 molecules. And it's usually bound to a negatively charged protein inside the vesicle. For neurons that need to fire incredibly fast, the body even has mechanisms for rapidly recycling these vesicles, ensuring a constant supply is ready to go. These vesicles have specific docking proteins on their surface, VMPs, which help line them up perfectly with the release sites on the inside of the nerve cell membrane.
Okay, lined up and ready. Then comes the big moment, release.
Yes, release. This is a critical step involving a complex molecular dance of proteins we collectively call snare proteins. Think of them as the molecular machinery that precisely aligns the vesicle and then fuses it with the nerve terminal membrane. Physiologic release is entirely dependent on calcium ions. When an electrical signal, an action potential, arrives at the nerve terminal, it triggers the opening of specialized N-type calcium channels. Calcium rushes into the nerve ending. This incoming calcium then interacts with a specific VMMP protein called synaptotagmin on the vesicle. This interaction triggers the fusion of the vesicle membrane with the terminal membrane, opening a pore and expelling the HEA out into the synaptic cleft. Now, a somatic motor nerve might release hundreds of these quanta with a single impulse. Autonomic postganglionic varicosities tend to release fewer, but often over a wider area. And frequently, other co-transmitters are released right alongside the HA, adding layers to the communication.
Okay, this is exactly where our opening case study comes alive. It reveals the precision of pharmacology, right? This detailed process of neurotransmitter release. This is precisely how botulinum toxin BONT works its magic. It blocks H vesicle release. How? By enzymatically cleaving, literally sniffing apart 2 amino acids from one or more of those critical snare fusion proteins we just talked about. This surgical disruption at the molecular level results in muscle paralysis, which, in our case study, precisely resolved the woman's blepharospasm by paralyzing those tiny eyelid muscles. It's a targeted localized paralysis.
Exactly, and you contrasted it perfectly with tetanus toxin 1010. It also cleaves similar fusion proteins, but its primary target is different. It hits inhibitory neurons in the spinal cord. So instead of paralysis, you get severe generalized muscle spasm. Same mechanism, different target, opposite effect.
And the remarkable thing is that a single treatment of botulinum toxin can provide relief for a significant period, typically 1 to 3 months.
Amazing precision. Okay, so HGH is released, it acts. How does its action stop? Right, the final step. Termination of action. The work of ASIC in the synapse is ended incredibly rapidly by an enzyme called acetylcholinesterase, or ATE. This enzyme is extremely efficient. It splits ATIC into choline and acetate, neither of which has any significant transmitter effect on its own. Most cholinergic synapses are packed with ATIC, meaning ATIC's half-life in the synaptic cleft is astonishingly short, just a fraction of a second. This ensures the signal is brief and precise. There are other cholinesterases, like butyrylcholinesterase found in plasma and other tissues, but ATIC is the primary cleanup crew right there at the synapse. Now, related to termination, let's talk about another crucial aspect of neurotransmission, how the body efficiently manages leftover neurotransmitters through specialized uptake carriers or transporters. There are two major families of these transport proteins. You have the ABC, or ATP binding cassette transporters, which use cellular energy ATP to move molecules. And then there are the SLC, or solute carrier transporters. These mostly harness existing ion gradients, usually sodium gradients, for cotransport. Sometimes they even transport substances in reverse out of the cell.
Okay, and these SLC transporters, they're incredibly relevant to pharmacology, aren't they? They seem to be key drug targets. You mentioned NET, the norepinephrine transporter, its scientific name being SLC6A2. That brings norepinephrine back into the nerve ending. Then there's D8, the dopamine transporter, SLC6A3 for dopamine reuptake, and SERT, the serotonin transporter, SLC6A4 for serotonin reuptake. They're like the cell's sophisticated recycling machines.
Exactly. And their importance extends directly into pharmacology. For instance, NET is crucial in mediating the peripheral actions of drugs like cocaine and amphetamines. They block it. In the CNS, NET, SERT, and D8 are critical targets for several classes of antidepressant drugs, and they're also central to the potent actions of recreational drugs like cocaine and amphetamines. We also see GABA transporters like GAT1, GT2, GA3. GA3 specifically is a target for certain anti-seizure medications. And glutamate, the major excitatory transmitter in the CNS, essential for learning and memory, relies on its own family of SLC transporters, the EAAT family. Understanding these transporters is absolutely fundamental for designing and understanding many modern drugs.
Right. Understanding the cleanup crew is as important as understanding the signal itself. Okay, let's shift focus now to the other major player, adrenergic neurons. How does norepinephrine go through its life cycle?
Okay, shifting to adrenergic neurons, the process of synthesis, storage, and release is similarly intricate, but with different players. Let's start with synthesis. First, the amino acid tyrosine is taken up into the nerve ending. Then it's converted to another molecule called DOPA by an enzyme called tyrosine hydroxylase. Now this step, the tyrosine hydroxylase step, is the rate-limiting step. It's the slowest, most controlled part of the whole synthesis process. And it can be specifically inhibited by the drug metazine. From DOPA, other enzymes then synthesize the final catecholamine transmitters, which could be dopamine, norepinephrine, or epinephrine, and these are then stored in membrane-bound vesicles. In most sympathetic postganglionic neurons, norepinephrine is the final product. Epinephrine formation happens mainly in the adrenal medulla and a few brain areas, and dopamine is the end product in specific dopaminergic neurons.
Okay, so synthesis is tightly controlled. What about storage?
For storage, there's a high-affinity loading dock called the vesicular monoamine transporter, or VMAT. This actively transports the catecholamines, dopamine and epi, into their storage vesicles. And this VMAT transporter can be inhibited by drugs like the resorpine alkaloids that includes resorpine itself, tetrabenazine, and deutetrabenazine. Blocking VMAT causes a significant depletion of transmitter stores within the nerve ending because it can't be properly packaged.
Makes sense. And release, similar to AS.
Then comes release. Yes, it's a calcium-dependent process, very similar mechanistically to what we described for cholinergic terminals. An action potential arrives, calcium rushes in through voltage-gated channels, and the vesicles fuse with the membrane, expelling their contents into the synaptic cleft. And importantly, along with norepinephrine, other crucial cotransmitters like ATP and the enzyme dopamine hydroxylase, which is involved in any synthesis, are often simultaneously released from the same vesicles, adding to the signaling complexity.
Okay, now here's another really interesting interaction, a deep dive highlight, I think. Indirectly acting sympathomimetics. You mentioned drugs like tyramine found in things like aged cheeses and wines, plus amphetamines and ephedrine. These can release stored transmitters from neuroadrenergic nerve endings, but you said it's by a calcium-independent process. How does that work? They don't need the electrical signal.
That's exactly right. It's quite clever, or maybe sneaky, depending on your perspective. These drugs aren't very good at activating the adrenergic receptors themselves. They're poor agonists, but they are excellent substrates for the monoamine transporters, especially NET, the norepinephrine transporter. So they get avidly taken up into the noradrenergic nerve endings by NET, basically hitching a ride on the nerve's own recycling system.
They trick the transporter.
Precisely. Once inside the nerve cytoplasm, they are then transported by VMAT into the storage vesicles. Inside the vesicle, they literally displace norepinephrine from its storage sites. This displaced norepinephrine then gets expelled out into the synaptic space, not by normal vesicle fusion, exocytosis, but by essentially running the NET transporter in reverse. It gets pushed out through NET. Amphetamines add another layer because they also inhibit monoamine oxidase, MAO, the enzyme that breaks down NE inside the neuron so more NE sticks around. And they don't strictly require vesicle exocytosis for their action. It's a sophisticated hijacking of the neuron's own machinery.
Wow, that's complex. Okay, so NE is released either normally or via these indirect actors. How is its action terminated?
Right. The termination of action for norepinephrine involves two main processes. First, there's metabolism. NE and epinephrine are broken down by two key enzymes, catechol O-methyltransferase, or COMT, and monoamine oxidase, or MAO. MAO is quite active even inside the resting nerve terminals, so there's a significant ongoing turnover of norepinephrine. We can actually measure the levels of metabolites like VMA and metanephrines, often in a 24-hour urine collection, to get an estimate of the body's overall catecholamine turnover.
Okay, so metabolism happens, but is that the main way the signal stops at the synapse?
Good question. While metabolism is important eventually, it isn't the primary mechanism for culminating the action of physiologically released norepinephrine right there at most synapses. The main mechanisms are actually simpler. First, just simple diffusion away from the receptor site, with eventual metabolism happening later in the plasma or liver. And second, crucially, reuptake back into the nerve terminal by NET, that transporter we just discussed, also known as uptake 1 or reuptake 1. Some reuptake also happens into nearby glial cells or other cells. NET acts like a powerful vacuum cleaner, sucking the NE back up very efficiently. And as we mentioned, this NET transporter is the specific target inhibited by drugs like cocaine, solriamfetol, and certain antidepressants. Blocking reuptake significantly increases the amount of active transmitter in the synaptic cleft and prolongs its effects.
Got it. Reuptake is key for any termination.
Absolutely. Now, it's really crucial to understand that beyond just the primary transmitters, we focused on ACE, SHE, and NE co-transmitters are also present in the vesicles of both cholinergic and adrenergic nerves. They can be found right there in the same vesicles as the primary transmitter, or sometimes even in separate populations of vesicles within the same nerve ending.
And what do these co-transmitters do? Are they just backups?
Oh, far from it. They play incredibly diverse and vital roles. They can provide faster or sometimes slower actions that supplement or modulate the effects of the primary transmitter, making the overall signal much more nuanced. They also participate in critical feedback inhibition loops, finely tuning the activity of the same nerve terminal or nearby nerve terminals. Think of it like a finely tuned orchestra. The main melody might be played by acetylcholine or norepinephrine, but then you have dozens of other instruments at the co-transmitters adding harmonies, different rhythms, subtle color, and emphasis.
Can you give some examples?
Sure. For instance, adenosine triphosphate, or ATP, often acts as a fast-acting, excitatory co-transmitter alongside the primary one at many nerve effector synapses. Nitric oxide, NO, is another key player. It's synthesized on demand, often triggered by cholinergic nerves, and it's a really important inhibitory co-transmitter, particularly for relaxing, smooth muscle like in blood vessels or sphincters in the gut. Then you have a whole range of peptides, things like vasoactive intestinal peptide, or VIP, which can act as a vasodilator or cardiac stimulant, or neuropeptide Y, NPY, which can inhibit fluid secretion in the gut and cause vasoconstriction. Substance P is important in sensory transmission. Calcitonin gene-related peptide, CGRP, is involved in cardiovascular sensory nerves. The list goes on.
Wow, it's not just one signal, it's a whole chord of signals being sent.
Exactly. And the sheer number of these co-transmitters, plus the fact that neuron growth and even which transmitters get expressed, can be dynamically influenced by things like neurotrophic factors or even environmental cues like the light-dark cycle. It just highlights the incredible adaptability and complexity of this system.
Okay, that's an incredible symphony of signals. So we've talked about the chemical keys, the neurotransmitters, and all these co-transmitters. Now let's talk about the specific locks these keys fit into. The receptors. You mentioned earlier that scientists define different receptor subtypes historically based on how drugs affected them, like muscarinic versus nicotinic for HEC or alpha versus beta for NE. But now, molecular biology gives us a clearer picture.
Precisely. Historically, it was pharmacology using specific agonist drugs that activate receptors and antagonist drugs that block them that allowed us to differentiate subtypes. But now, with molecular biology, we can actually clone and express the specific genes for these receptors. This lets us study them in isolation and understand their structure and signaling pathways much more precisely. So, let's start with cholineceptors, the ones that respond to acetylcholine. The two primary types are named after the alkaloids that historically help distinguish them: muscarine from certain mushrooms and nicotine from tobacco. First, we have muscarinic cholineceptors, usually labeled M1 through M5. These are all G protein-coupled receptors, meaning they trigger intracellular signaling cascades when activated. M1, M3, and M5 receptor activation typically leads to the formation of signaling molecules inside the cell, like IP3 and DAG, which then cause an increase in intracellular calcium levels. This calcium increase drives various cellular responses, often excitatory. Where do you find them? Well, M1 receptors are important in CNS neurons, on sympathetic postganglionic neurons receiving cholinergic input, and at presynaptic sites. M3 receptors are crucial for stimulating exocrine glands, like salivary glands, influencing blood vessels, often causing relaxation via endothelium, and they also have roles in the CNS. M5 receptors are found in vascular endothelium and the CNS as well.
Okay, so M1, M3, M5 generally get things going via calcium. What about M2 and M4?
M2 and M4 receptors work differently. Their activation generally results in the opening of potassium channels and the inhibition of an enzyme called adenyl cyclase. This leads to slower, often inhibitory, effects like slowing the heart rate. M2 receptors are found prominently in the myocardium, heart muscle, in smooth muscle, at presynaptic nerve terminals, acting as autoreceptors to inhibit ACE release, and also in the CNS. M4 receptors are mainly located in the CNS, possibly also on vagal nerve endings.
Got it. Muscarinic are G protein coupled with different downstream effects. What about the other type, nicotinic?
Then we have nicotinic cholineceptors, labeled NN neuronal type and NM muscle type. These are fundamentally different from muscarinic receptors. They are ligand-gated ion channels. This means when acetylcholine binds directly to the receptor itself forms a channel that rapidly opens, allowing ions, primarily sodium and some potassium, to flow across the membrane. This ion flow causes a rapid depolarization of the cell, which can quickly trigger an action potential if the depolarization reaches threshold. It's a much faster, more direct effect than the G protein-coupled muscarinic receptors.
Faster, direct excitation. Where are these found?
NN receptors are found on the cell bodies of postganglionic neurons in all autonomic ganglia, both sympathetic and parasympathetic. They're also found on some presynaptic cholinergic terminals where they can enhance AC release. They are essential for transmitting the signal from the preganglionic to the postganglionic neuron. NM receptors are specifically located at the skeletal muscle neuromuscular end plates. These are the receptors that mediate voluntary muscle contraction when stimulated by somatic motor nerves.
Okay, that clarifies the A2A receptors. Now, what about the adrenoceptors, the ones responding to norepinephrine and epinephrine?
Right. For adrenoceptors, the general term describes receptors that respond to these catecholamines. These are also G protein-coupled receptors, and they are subdivided into alpha, further split into Au and Av-mer, split into G-point, Ver, and Vale subtypes. And we also need to include dopamine receptors, D1, D5 here, as dopamine is a precursor and a transmitter itself.
Alpha and beta. Okay, let's start with alpha.
Alpha-1 receptors are typically found in postsynaptic effector cells, especially smooth muscle cells like those in blood vessels. Their activation, similar to M1, M3, M5, generally leads to IP3 and DAG formation, increasing intracellular calcium and usually causing contraction like vasoconstriction. Alpha-2 receptors have a different profile. They are very often found on presynaptic adrenergic nerve terminals. Here, they act as crucial inhibitory autoreceptors. When NE binds to them, it inhibits further NE release from that same terminal. It's a key negative feedback mechanism. Alpha-2 receptors are also found on some other cells like platelets, fat cells, lipocytes, and some smooth muscle. In these locations, their activation generally leads to inhibition of adenyl cyclase, reducing intracellular KMP levels.
Okay, I1-Win usually excites. IGIA2 often inhibits it, especially presynaptically. What about the beta receptors?
Beta-adrenoceptors generally work by stimulating the enzyme adenyl cyclase, which increases intracellular levels of cyclic AMP, CMP, another important signaling molecule. Beta-1 receptors are predominantly located in the heart. Their stimulation increases heart rate, contractility, force of contraction, and conduction velocity. They're also found in fat cells, the brain, some presynaptic sites, the juxtaglomerular apparatus in the kidney, involved in renin release and blood pressure control, and the ciliary body epithelium in the eye, involved in aqueous humor production. Beta-2 radi-2 receptors are found widely on smooth muscle, particularly in the airways, bronchioles, blood vessels supplying skeletal muscle, and the uterus. Activation here typically causes relaxation of the smooth muscle, so bronchodilation and vasodilation in those specific beds. They're also found on cardiac muscle, where they can also increase heart rate and contractility, though beta-1 effects usually dominate in the heart. Beta-3 receptors are primarily found in leukocytes, fat cells, where they are involved in stimulating lipolysis, fat breakdown. They are also present in the heart and possibly the bladder, also stimulating adenyl cyclase.
So beta receptors generally stimulate CAMP production, leading to different effects depending on the tissue: heart stimulation via DM1, smooth muscle relaxation via beta-2,
Interesting.
and dopamine receptors.
And finally, dopamine receptors, D1, D5. These are also G protein coupled and are critical in both the CNS and the periphery. D1-like receptors, D1 and D5, typically stimulate adenyl cyclase, increasing CAMP. They are found in the brain and, importantly, in the smooth muscle of renal kidney blood vessels, where their activation causes vasodilation, increasing blood flow to the kidneys. D2-like receptors, D2, D3, D4, generally inhibit adenyl cyclase, decreasing CAMPAC. They are found in the brain, some smooth muscle, and importantly, on presynaptic nerve terminals where they can inhibit transmitter release. D2 receptors can also increase potassium conductance, leading to hyperpolarization. Understanding these specific receptor subtypes, their locations, and their signaling mechanisms is absolutely critical for mastering how drugs can selectively target them to achieve desired therapeutic effects, or sometimes cause unwanted side effects.
Absolutely critical. Okay, so we have the classic classifications: cholinergic and adrenergic, with all their receptor subtypes. But what about the nonconformists? You mentioned earlier that for years researchers recognized these non-adrenergic, non-cholinergic, or NANC neurons in places like the gut, airways, bladder. They just didn't seem to fit the standard ATH or NE profile.
That's right. Both motor and sensory NANC fibers exist in these tissues, and they represent a really significant portion of the autonomic innervation, especially in places like the gut. While peptides are probably the most common transmitter substances found in these NANC nerve endings, other important signaling molecules are also crucially involved. Things like nitric oxide synthase, the enzyme that produces a nitric oxide, NO, and purines like ATP acting as a transmitter are key players.
And you mentioned capsaicin earlier, the stuff in chili peppers.
Yes, interestingly, capsaicin can cause selective release of transmitters, especially the sensory peptide substance P, from these NANC sensory neurons. And if you use high doses, it can even selectively destroy these types of neurons, which highlights their unique chemical sensitivity compared to classic cholinergic or adrenergic nerves.
Wow. So where are these NANC systems most prominent?
The enteric nervous system, that brain in the gut we talked about, is by far the most extensively studied NANC system. It contains these NANC neurons alongside its cholinergic and adrenergic components. In the small intestine, for example, individual NANC neurons can contain a truly rich cocktail of signaling molecules. You might find one or more of these in a single neuron: nitric oxide synthase, making NO, calcitonin gene-related peptide, CGRP, cholecystokinin, CCK, dynorphin, an opioid peptide, and kephalins, other opioid peptides, gastrin-releasing peptides, serotonin, 5-HT, neuropeptide Y, NPY, somatostatin, substance P, and vasoactive intestinal peptide, VIP. It's really not uncommon for a single enteric neuron to contain as many as 5 different transmitters. This allows for an incredibly nuanced and complex level of local communication within the gut wall.
5 different transmitters on one neuron. That's incredible complexity. And you mentioned sensory NANC fibers too. How they work differently?
Yes, these NANC sensory fibers are often described using terms like sensory efferent or sensory local effector fibers. This is because they have a dual role. When they're activated by a sensory input, like stretch or a chemical stimulus in the gut, they don't just send a signal towards the CNS. They can also release transmitter peptides directly from the sensory ending itself, or from local axon branches, and even from collaterals that terminate back in the autonomic ganglia. These released peptides, like substance P or CGRP, are often potent agonists, activators, in many autonomic effector tissues. So the sensory fiber itself can directly cause a powerful, localized response right where the stimulus is being sensed. It's a very efficient local reflex loop.
A local alarm and response system built right in.
Exactly. Ultimately, mastering autonomic function really means understanding that it's integrated and regulated at multiple levels. It's not just one switchboard. It runs from the highest levels of the central nervous system all the way down to the individual effector cells. And this regulation primarily relies on sophisticated negative feedback loops, creating a constant adjustment system, though other complex mechanisms are definitely also involved.
Okay, so let's talk about that integration. At the highest level, in the midbrain and medulla, the two main ANS divisions, sympathetic and parasympathetic, are constantly being integrated, right? Along with input from the endocrine system, sensory input from all over the body, and information flowing down from higher CNS centers like the cerebral cortex. Early investigators had those descriptive terms. They called the parasympathetic system trophotropic, kind of like rest and digest, focused on energy conservation, slowing the heart, stimulating digestion. And in contrast, the sympathetic system was aptly called ergotropic, more like fight or flight, geared for energy expenditure, think cardiac stimulation, increased blood sugar, sending blood away from the skin, cutaneous vasoconstriction. It's that fundamental balance, isn't it?
It is, and there are also crucial cooperative interactions happening at more subtle levels, precisely tuned within the brainstem, medulla, and spinal cord. For example, parasympathetic sensory fibers can exert powerful reflex control over sympathetic motor outflow. A classic illustration is how sensory fibers from the carotid sinus baroreceptors, the ones that detect changes in your blood pressure, have a major influence on sympathetic outflow coming from the vasomotor center in the brainstem. This helps regulate your blood vessel tone constantly. Similarly, within the gut, those ENS sensory fibers are constantly synapsing on motor neurons right there in the gut wall, creating incredibly complex localized reflexes without necessarily involving the brain directly for every little adjustment.
Right, it's layers upon layers of control. This brings us to another fantastic example of the ANS's brilliant compensatory power, and it's definitely a deep dive highlight. Cardiovascular integration. You often see this illustrated in diagrams, like figure 6 to 7 from the KATZUNG text. Mean arterial pressure? Your average blood pressure is the primary controlled variable that your body works tirelessly, moment to moment, to keep within a certain range. And what's truly fascinating and tells you a lot about baseline control is looking at what happens without autonomic control. Take a transplanted heart, for example, which has lost its original nerve supply. Its intrinsic resting heart rate is actually much higher, maybe around 110, 130 beats per minute.
That's right. Which immediately tells us that in a normal, intact, resting human, our typical heart rate of maybe 70, 80 BPM is actually being held down, dominated by continuous parasympathetic vagal tone. The vagus nerve is constantly applying the brakes at rest.
Exactly. So, now consider what happens if we give a slow intravenous infusion of norepinephrine to a normal person with all their reflexes intact. Norepinephrine itself has direct effects, right? It's a powerful vasoconstrictor, so it dramatically increases peripheral vascular resistance, which raises mean arterial pressure, MAP. If there were no reflexes, like in that transplanted heart, it would also directly stimulate the heart itself, increasing both heart rate and the force of contraction.
But in a normal subject with intact reflexes, that rising MAP immediately triggers a powerful negative feedback loop. The increased pressure is sensed by those baroreceptors in the carotid sinus and aortic arch. Their firing rate increases, sending signals to the brainstem. The result: a decrease in sympathetic outflow to the heart, and simultaneously a powerful increase in parasympathetic, or vagus nerve, discharge directed right at the SA node, the heart's natural pacemaker.
So the body fights back against the drug's direct effect on the heart.
Precisely. The net effect you observe in a normal person getting that NE infusion is a marked increase in peripheral vascular resistance and an increased MAP due to the direct vasoconstriction, but often, quite counterintuitively, a slowing of the heart rate bradycardia. This reflex compensatory response is directly opposite to the drug's direct stimulating action on the heart muscle itself, yet is completely predictable once you understand the integrated cardiovascular regulation performed by the ANS. It's truly a masterclass in biological feedback control.
It really is. And this principle of feedback, it also extends right down to the nerve endings themselves, doesn't it? You mentioned presynaptic regulation.
Yes, this is a vital part of fine-tuning neurotransmitter release at the source. At most nerve endings, there are important presynaptic feedback control mechanisms. These often involve the transmitter itself influencing its own release. A really well-documented example is the alpha-2 receptor found right there on noradrenergic nerve terminals. When it gets activated by norepinephrine, the very transmitter being released from that terminal, this receptor triggers a signaling cascade that diminishes further any release. It usually does this by inhibiting calcium influx, which, as we discussed, is the crucial trigger for vesicle fusion and transmitter expulsion.
So any release inhibits further any release, a built-in brake.
Exactly. Conversely, sometimes you find presynaptic receptors that facilitate release. For instance, a presynaptic beta receptor on some adrenergic neurons can actually enhance any release when stimulated. These receptors that respond to the primary transmitter released by the nerve ending itself are called autoreceptors, and they're usually inhibitory, like the AUTO2 example, but occasionally they can be excitatory, like the presynaptic or certain nicotinic receptors on somatic motor nerve terminals.
Okay, autoreceptors respond to the nerve's own transmitter. But you also mentioned heteroreceptors.
Right. Nerve terminals don't just listen to themselves. They also have regulatory receptors that respond to substances released from other nearby nerve terminals. These are called heteroreceptors. For example, we mentioned that parasympathetic vagal fibers in the heart can synapse directly on sympathetic noradrenergic nerve terminals and release ASG, which then acts on muscarinic heteroreceptors on the sympathetic terminal to inhibit norepinephrine release. It's like crosstalk between the two systems right at the synapse. Alternatively, the ligands, the molecules that bind for these heteroreceptors, might just diffuse over from the blood or nearby tissues. Things like angiotensin II or certain prostaglandins circulating in the tissue fluid can bind to heteroreceptors on nerve terminals and modulate transmitter release, either increasing or decreasing it. This kind of presynaptic regulation by various endogenous chemicals, neurotransmitters, hormones, local mediators likely occurs at virtually all synapses, providing an incredibly exquisite layer of fine-tuning for transmitter release, moment by moment.
Layers upon layers of control. Then there's also postsynaptic regulation, right? How the receiving cell adjusts its own sensitivity over time.
Absolutely. The target cell isn't just a passive recipient. Its responsiveness can change based on its recent history of activation. This involves modulation by previous activity at the primary receptor. This can lead to either upregulation, where the cell becomes more sensitive, perhaps by increasing the number of receptors on its surface, typically occurring after a period of decreased activation, or downregulation, where the cell becomes less sensitive, perhaps by decreasing receptor numbers or uncoupling them from signaling pathways, usually happening after prolonged or intense activation.
And there's that extreme form, denervation, supersensitivity.
Yes, that's a dramatic example of upregulation. It happens after a tissue is completely deprived of its nerve supply, perhaps due to injury. For instance, in normal skeletal muscle, those nicotinic receptors, NM-type, are tightly clustered right at the nerve end plate. But if you cut the motor nerve, these receptors dramatically proliferate and spread out across the entire surface of the muscle fiber. This makes the muscle exquisitely sensitive to even tiny amounts of acetylcholine that might reach it through the circulation.
And this can happen with drugs too. Pharmacologic supersensitivity.
Right. A similar, though usually less dramatic, supersensitivity can occur if you use drugs that prevent receptor activation for a long time. For example, giving a drug like reserpine, which depletes norepinephrine stores, effectively silences adrenergic transmission. If you do this for a prolonged period, the postsynaptic tissues can become supersensitive to norepinephrine. Then, if you suddenly introduce any or an agonist, you might see an exaggerated response. This principle helps explain why the effects of some drugs can change over time with chronic use, or why withdrawal effects occur. The system adapts.
Okay, now let's look at another deep dive highlight that really pulls together multiple concepts. Complex ganglionic transmission. You mentioned this involves a sequence of potentials, not just a simple on signal. Can you walk us through that again? It sounds like a perfect illustration of subtle, layered interactions fine-tuning the response.
It really is. It's often depicted in diagrams, like figure 6-8 from KATZUNG, showing the electrical events in a postganglionic neuron after its preganglionic input fires just once. You don't just see a single switch flicking on. Instead, there's this fascinating sequence of electrical potentials recorded inside the postganglionic cell. First, and most prominently, there's a very rapid, large depolarization called the fast excitatory postsynaptic potential, or fast ETSP. This is caused by Hc activating those ligand-gated neuronal nicotinic and N receptors we talked about. It's usually strong enough to make the postganglionic neuron fire its own action potential, passing the signal along quickly.
Yeah. Okay, the main signal gets through fast via nicotinic receptors, but then other things happen.
Yes. This fast EPSP is often immediately followed by a small, but much slower and longer-lasting hyperpolarization. The cell's membrane potential dips below resting level. This is called the slow inhibitory postsynaptic potential, or slow IPSP. This hyperpolarizing effect involves HRS acting on muscarinic M2 receptors on the same postsynaptic neuron, which open potassium channels, letting positive charge flow out and making the cell temporarily less likely to fire again right away.
Interesting. So an initial fast go signal followed by a slower hold on a sex signal.
Kind of, but it doesn't stop there. Following the slow IPSP, there's often another small, slow, but this time excitatory postsynaptic potential, a slow EPSP. This one is thought to be mediated by ACHRED acting on muscarinic M1 receptors, which work by closing certain potassium channels, reducing potassium outflow, and thus making the cell slightly more excitable again for a period.
Wow. Fast on, slow off, slow on again.
And sometimes there's even a fourth potential observed, a late, very slow EPSP, which can last for minutes. This one is potentially evoked by peptides, those co-transmitters, released perhaps from the same preganglionic terminal or other nearby fibers, acting on their own specific receptors. The key point here is that these slower potentials, the slow IPSP, the slow EPSP, the late slow EPSP, they aren't primarily about triggering immediate firing. Rather, they subtly modulate the overall responsiveness or excitability of the postsynaptic neuron to subsequent incoming primary excitatory activity, those fast EPSPs. They allow for incredibly nuanced, context-dependent, precise control over how readily the signal gets passed through the ganglion. It's sophisticated tuning.
That is incredibly sophisticated. So many intricate layers just to get a signal across a single synapse reliably and with modulation. But this really deep understanding of ANS transmission, all these synthesis, storage, release, termination steps, all the receptors, the co-transmitters, the feedback loops, this is precisely what allows for such selective pharmacologic modification, isn't it? And it also explains why some drugs, maybe acting on broader mechanisms, can unfortunately have those non-selective side effects scattered throughout the body.
Absolutely. Let's systematically walk through how drugs target these precise steps in autonomic transmission. This is essentially the pharmacologist's toolkit, and we can reference insights often summarized in tables like Table 6 to 5 in pharmacology texts. First, thinking really broadly, you can target action potential propagation itself. This is usually non-selective, though. Drugs like local anesthetics, like lidocaine, or potent natural toxins, like tetrodotoxin from pufferfish, and saxitoxin from certain shellfish, work by blocking voltage-gated sodium channels, which are essential for the electrical signal to even travel down the nerve axon. Block the channels, block the signal transmission entirely.
Okay, stopping the signal before it even gets to the terminal. What about interfering with the transmitter itself? Synthesis?
Exactly. You can target transmitter synthesis. At cholinergic terminals, we mentioned the hemicoliniums. They specifically block the CHT transporter responsible for choline uptake, thereby slowing down AK synthesis because a key ingredient is missing. At adrenergic terminals, or in the adrenal medulla, the drug alpha-methyltyrosine, metarosine, inhibits the enzyme tyrosine hydroxylase. Since that's the rate-limiting step, this effectively blocks the synthesis of all catecholamines. Dopamine, NE, epinephrine.
Okay, block synthesis. Or you could mess with storage.
Yes, transmitter storage is another key target. For cholinergic vesicles, the drug visamicol blocks the VAT transporter that loads HEA into vesicles. HE gets made, but it can't be stored properly, leading to depletion of readily releasable transmitter over time. For adrenergic vesicles, drugs like reserpine and tetrabenazine block the VMAT transporter. This prevents dopamine and norepinephrine from being loaded into vesicles, leaving them vulnerable to breakdown in the cytoplasm and leading to profound depletion of catecholamine stores.
Synthesis, storage, then release itself must be a major target area.
Definitely. Transmitter release is a really rich target for pharmacological intervention. As we discussed, many presynaptic receptors, autoreceptors, and heteroreceptors for things like norepinephrine, dopamine, acetylcholine, angiotensin II, and prostaglandins can modulate release, either enhancing or inhibiting it, and drugs can be designed to activate or block these specific receptors. We also have toxins that directly interfere with the release machinery. Conotoxin GVIA from a marine snail potently reduces transmitter release by specifically blocking the N-type calcium channels that are crucial for triggering exocytosis. Conversely, the drug amifampridine actually increases transmitter release. It works by blocking certain potassium channels in the nerve terminal, which prolongs the duration of the action potential, allowing more calcium to enter and thus enhancing vesicle fusion. Domoic acid, another toxin, can modulate release via kainate receptors, though this is more significant in the CNS. And of course, revisiting our star example, botulinum toxin. It specifically prevents A release from cholinergic vesicles by enzymatically cleaving those essential snare proteins involved in fusion.
The ultimate blocker of release, at least for HIF.
For ATIS release, yes. At the other extreme, you have elatrotoxin, the venom from black widow spiders. It causes a massive, explosive, uncontrolled release of transmitter from both cholinergic and adrenergic terminals, leading to system overload. And then we have the indirectly acting drugs like tyramine and amphetamine, which promote adrenergic transmitter release through that calcium-independent displacement mechanism we detailed earlier.
Okay, lots of ways to affect release. What about after release? The reuptake process.
Transmitter reuptake after release is another major target, particularly for adrenergic neurons. Drugs like cocaine, many tricyclic antidepressants like imipramine, and the SNRI class of antidepressants like venlafaxine work by inhibiting NET, the norepinephrine transporter located on the adrenergic nerve terminal membrane. By blocking this reuptake vacuum cleaner, they increase the concentration and duration of action of norepinephrine in the synaptic cleft, thus boosting its effect on postsynaptic receptors. Similar drugs target the dopamine DAT and serotonin SERT transporters.
Right, and then perhaps the most selective approach is directly hitting the receptors.
Exactly. Targeting receptor activation or blockade generally offers the maximum potential for selectivity because the different receptor subtypes are often located in specific tissues and mediate specific functions. So, for example, norepinephrine itself is an agonist that activates IN-MAC1 receptors primarily. Phentolamine is an antagonist that blocks I receptors, both IL-1 and AD2. Isoproterenol is a potent agonist primarily for I receptors, COL1 and A2. Propranolol is a classic antagonist that blocks I receptors, both I1 and A2. For cholinergic receptors, nicotine is the classic agonist for nicotinic receptors. Tubocurarine, derived from curare, is an antagonist that blocks NM receptors at the neuromuscular junction. Bethanocol is an agonist that selectively activates muscarinic receptors, and atropine is a powerful antagonist that blocks muscarinic receptors throughout the body.
So many specific keys and locks to target. And one last step, breaking down the transmitter.
Yes, interfering with enzymatic inactivation of transmitter is the final major strategy. For acetylcholine, drugs like neostigmine and physostigmine inhibit the enzyme acetylcholinesterase, ACE. By preventing the breakdown of ESO, they prolong its action at cholinergic synapses, effectively amplifying the signal. For catecholamines, drugs like tranylcypromine inhibit the enzyme monoamine oxidase, MAO, which is involved in breaking down NE and dopamine inside the nerve terminal and elsewhere. Inhibiting MAO increases the amount of transmitter stored in vesicles, potentially leading to enhanced release. Each of these 5 or 6 steps, synthesis, storage, release, reuptake, receptor interaction, degradation represents a distinct point where drugs can intervene to modify autonomic function, allowing for highly targeted therapeutic effects if used correctly.
That's a fantastic overview of the pharmacologist's toolkit. Let's try to bring this all together now with a really practical, maybe everyday example that listeners might have experienced or at least observed. The pharmacology of the eye. The eye seems like a perfect miniature model system because it clearly demonstrates multiple distinct autonomic functions, each controlled by various specific receptors we've just discussed.
It really is an excellent example. Let's break it down by the 2 divisions. First, consider parasympathetic control of the eye. The pupillary constrictor muscle, that's the circular muscle in your iris that makes your pupil smaller, is richly supplied with muscarinic receptors, specifically M3 type. When these M3 receptors are activated by AA, released from parasympathetic nerves, the muscle contracts, causing miosis, a reduction in pupil size. You'd see this effect with the drugs that mimic AA cholinomimetics or drugs that block ACE, like certain organophosphate insecticides or nerve agents, causing pinpoint pupils.
Okay, parasympathetic constricts the pupil via M3. What else?
The ciliary muscle, which is inside your eye and controls the shape of your lens for focusing, also has M3 muscarinic receptors. When it contracts, it allows the lens to become more convex, enabling accommodation for near vision focusing on close objects. Strong contraction can lead to cyclospasm or ciliary muscle spasm. Now, crucially for glaucoma treatment, this contraction of the ciliary muscle also puts tension on a structure called the trabecular meshwork, which is like a filter near the edge of the iris. This tension opens up the pores in the meshwork, facilitating the outflow of aqueous humor, the fluid inside the front part of your eye, into a drainage channel called the canal of Schlemm.
Ah, so contracting the ciliary muscle helps drain fluid and lowers eye pressure.
Exactly. This reduction in intraocular pressure is a key therapeutic strategy for managing glaucoma. So all these parasympathetic effects, the miosis, the accommodation or cyclospasm, and the reduced eye pressure due to enhanced outflow are all mediated by M3 receptors and are prevented or reversed by muscarinic blocking drugs like atropine, which would cause pupil dilation, paralysis of accommodation, and potentially increase eye pressure in susceptible individuals.
Got it. That's the parasympathetic side. Now, what about sympathetic control of the eye?
Okay, for sympathetic control, the pupillary dilator muscle, this is the radial muscle in the iris, running outwards like spokes on a wheel, has alpha-1 receptors. When these alpha-1 receptors are activated by norepinephrine, released from sympathetic nerves, this muscle contracts, pulling the iris open and causing mydriasis and increase in pupil size. This happens naturally during sympathetic discharge, like when you're startled or in dim light. It can also be induced pharmacologically with alpha-1 agonist drugs like phenylephrine, which ophthalmologists often use in eye drops to dilate pupils for an eye exam.
Okay, sympathetic dilates the pupil via IGO1. Does it affect pressure too?
Yes, but through a different mechanism. The ciliary epithelium, which is the tissue that actually produces the aqueous humor fluid in the first place, has beta receptors, likely ACO1 and BA2. Activating these beta receptors seems to facilitate the secretion of aqueous humor. So strategically, if you want to lower intraocular pressure for glaucoma, you can block these beta receptors. Beta-blocking eye drops like Timol reduce the production secretion of aqueous humor by the ciliary epithelium, thereby lowering intraocular pressure. This provides another very effective therapy for glaucoma, working by a completely different mechanism than the parasympathetic acting drugs that enhance outflow.
Wow. So you have parasympathetic controlling constriction and outflow via M3, and sympathetic controlling dilation via AO1, and production via AOA. It really is a perfectly choreographed dance of different receptors and muscles, all controlled by the ANS, right there in the eye. Okay, wow. We've truly gone deep today, haven't we? I mean, from the broadest divisions of the nervous system all the way down to the intricate dance of specific neurotransmitters, their unique receptors, and precisely how various drugs can strategically intervene at each and every step. I feel like you, our listener, should now have a really comprehensive, truly elite professional-level understanding of the autonomic nervous system's profound role in controlling our most vital, often completely unseen bodily functions. Mastering this depth of knowledge is really key.
Absolutely, and if we connect all these details back to the bigger picture, it becomes clear that mastering this information provides not just those aha moments, though hopefully there were a few of those, but it provides a robust framework for truly understanding human physiology at a deep level, and critically, how we can therapeutically influence it with remarkable precision when things go wrong. It just highlights the incredible precision, the redundancy, and the adaptability that's built right into our biological systems. It's quite humbling, really.
It really is. So maybe a final thought for you, our listener, as you go about your day. Consider how many of the everyday drugs we encounter, maybe drugs you take yourself, or friends or family take, even those for seemingly unrelated conditions like allergies or depression or high blood pressure. How might they be subtly or perhaps significantly interacting with these hidden controls of our autonomic nervous system, shaping our physical experiences, our side effects, our overall well-being in ways we rarely even consider? It's truly a hidden influence in pharmacology and daily life.
That's a great point. What really stands out to you now about this incredible system, this intricate balance between excitation and inhibition, between fight or flight and rest and digest, all happening automatically. The more you observe your own body and think about these mechanisms, the more questions inevitably arise.
Keep diving deep, keep asking those questions, and we'll see you next time on The Deep Dive.